Welcome to Click for IB Biology. Our topic is neural signaling, and here we're looking at the initiation of a nerve impulse. There's a blank version of my presentation. That would be a PDF that's linked into the description. You can download that, annotate with your own notes, and then why not take the 10-question quiz, which is also there with answers, so that you can check your understanding of this part of the course. So let's take a look at the initiation of a nerve impulse, and let's find our location to begin with. So the propagation of a nerve impulse means the transmission of the impulse along the axon, and as previously explained, this is called the action potential, and is actually measured at plus 30 millivolts with a millivolt meter, and that can be found to be diffusing along the axon here to the neuromuscular junction, where it would cause a muscle contraction. But our concern is, how does this actually begin when the resting potential is already sitting at minus 70 millivolts? So how do we get from minus 70 millivolts to plus 30 millivolts? And this begins with the interaction between cells of the central nervous system and our motor nerve, as shown in the image to the right. And that occurs at the synaptic junction between the nerve cell of the central nervous system and the motor nerve, and involves the soma, the cell body, and particularly the accumulation of these effects at what is called the axon hillock, just here in front of the beginning of the axon. So let's take a look at how this change in potential occurs. So this is the presynaptic membrane, which this is the cell of the central nervous system, and the postsynaptic membrane is the membrane of the motor neuron. There is a small gap between the central nervous system and motor neuron cell, and this is called the synaptic cleft, which is measured at a distance of between 20 to 40 nanometers, an incredibly short distance between the end of this nerve cell and the beginning of this motor neuron. The end of the cell, which is the cell of the central nervous system, contains a large number of vesicles that contain the transmitter substance, which is called acetylcholine. And this is stored within these vesicles. And this acetylcholine is the transmitter substance which will diffuse across the synaptic cleft to encounter the receptors in the motor neuron cell body. The release of this acetylcholine begins, step one, with a wave of sodium ions from the neuron in the central nervous system, going down its dendrite and axon towards the end of the synapse. This wave of sodium ions causes then an influx in calcium ions, 2+, into the synaptic structure at the end of the dendrite, and that causes the vesicles to migrate to the cell membrane. So 1, the sodium; 2, the activation of the calcium pumps; 3, the calcium ions cause the vesicles to migrate to the membrane where they fuse. Let's call that number 4. Point number 5 is the diffusion of the transmitter substance acetylcholine across the synaptic cleft, which, as previously mentioned, is an incredibly short distance and is almost instantaneous. And there they encounter receptors in the cell membrane of the soma of the motor nerve in the cell body. These receptors are called NACHR, acetylcholine receptors, nicotinamide acetylcholine receptors. And as the acetylcholine engages with these receptors, what happens is that they will allow sodium ions to enter the cell body here like this. Now, these sodium ions come from the extracellular fluid around the nerve cell. So that would be our 6th point. So let's review our progress. Sodium ions come in from the central nervous system dendrite. 2, calcium ions are pumped into the synaptic structure at the end. 3, the vesicles merge with the membrane. 4, the acetylcholine is now in the synaptic cleft across which it diffuses. 5, it engages with the receptor. We can name that receptor. And 6, this allows sodium ions to enter the cell body of the soma of the motor neuron. Now let's look at what's happening on the oscilloscope reading, which was sitting at minus 70 millivolts. But as we have added sodium ions, the recording in here of minus 70 millivolts will rise in a number of steps to reach minus 55 millivolts. And this value of minus 55 millivolts is called threshold. Now, threshold tells us that what's about to happen is what was generalized as an all or nothing event, and that if the charge arrives at minus 55 millivolts at the axon hillock, let's call this number 7, then we reach threshold, and then what happens is that the all is that an action potential is generated. The charge will go up radically and very, very quickly to plus 30 millivolts, like so. And that's the action potential, the nerve impulse, which we have been looking to explain the beginning of. And that would be occurring in this location here, like so. So this is how the nerve impulse is actually initiated. For higher level students, just a little bit more information about thresholds. The way in which the resting potential steps up to reach threshold can occur in essentially 2 different ways. They are known as spatial and temporal summation. In spatial summation, the postsynaptic soma is in contact with a number of different incoming nerves from the central nervous system, in my case here showing 3, and so each of those would contribute to the charge change from minus 70 millivolts to minus 55 millivolts threshold. So that's a spatial arrangement of different number of neurons. Another way in which this can occur is what's called a temporal summation, temporal referring to time. And the idea here is that there's a strong impulse coming along the axon of the presynaptic neuron, and that there are a number of stimulations of the postsynaptic neuron by these waves of transmitter substance across the cleft here like this. And so we only have a 1-to-1 correspondence of these 2 cells here. And so the rise going up like this is caused by 1 cell in the central nervous system, not by a number of. And this is called temporal summation. So 2 different ways in which this can be achieved, the action, the so-called threshold for the action potential. Now what we're going to do is we're going to go on and study how this action potential forms, and indeed how it is transmitted along the axon of a motor neuron. And that's in the next video, which is linked in here, along with the chance to subscribe to the site. And there's the playlist for you to get a quick reference for the different videos in this topic.